everyone to this Monco talk. And I will say just one thing beforehand about the Q&A session. So we will have about 10 minutes for questions after the talk. But you have, if you have urgent questions during the talk, it's also okay to ask immediately. So the speaker today is Professor Guo-Qiang Bi, invited by Professor Zhaoping Li. So Zhaoping, could you please introduce Professor Bi to the audience?
Okay.
Thank you.
Okay. Oh, thank you. Yeah. Thank you, Jingyu, for the organization facilitation very nicely. And we are very pleased and lucky to have Professor Guo-Qiang Bi available to give us a talk on this very exciting topic for our MOGO series. And MOGO seminar series is for the Max Planck Institute for Biological Cybernetics, of course, open to the whole Tübingen campus and worldwide Neuro campus. And people in neuroscience perhaps are not unfamiliar with Guo-Qiang Bi because of this very famous paper people probably know in the 1990s, Bi and Pu, on the spike timing dependent plasticity, which is one of these characteristic, amazing work, from learning to biophysics and how this old classical work of Hebbian learning could be in a new, more detailed, actually physiological and neural substrate with much more detail. In fact, is more than just more detail. Everybody see it in the textbook, so I'm not going to elaborate. And Professor Guo-Qiang Bi's latest work, in fact, I've heard some earlier version, maybe a few years ago, about this VISoR, but that was more focused on the like a hippocampus and reverberating circuits. And now this is fantastic, having the whole brain imaging in this such micron detail, and it's on monkeys, not just in rodents, and also in such a short time. So this is such a fantastic technology, and we're really looking forward to hearing it. And thank you very much, Guo-Qiang, for being available to present this for us. And Jingyu, you talk about that people could ask questions immediately during the talk. Is this okay with you, or shall we just have people maybe type it in the chat and ask later? What do you think, Guo-Qiang? What do you prefer?
Yeah, I think it's fine to ask to interrupt it.
Okay. So then we will just let people to do so then. And so I don't want to waste too much time. So without further ado, thank you very much, Guo-Qiang. Please go ahead. We'd like to hear from you. Yeah.
Thank you, Zhaoping. It's really grateful for Zhaoping and Jingyu also to invite me to give this talk. It's my honor to have this opportunity to share our recent work. I think it's mainly about some imaging methods trying to see the complex brain structures at primarily two extreme scales. One is on the left, it's a neuronal synapse that's less than 1 micron in size. It's imaged by cryo-electron tomography. With this approach, what we'd like to do is to solve the organization of the molecular machinery inside the synapse. And on the right is what Zhaoping just mentioned, that it's an entire monkey brain that imaged with our recently developed with high-speed 3D microscopy technique. We call this VISoR. So what we hope to use this for is to dissect the architecture of the neuronal circuits across the entire brain. And the work was primarily done by many very talented and dedicated students, postdocs, and colleagues in the lab, in my lab at USTC in China, and I have been very lucky to have them. And I want to start with this question: our brain understand how our brain works. I think Guo-Qiang several years ago had raised this similar topic in Beijing in this future forum that unfortunately I wasn't able to attend. But I remember to show this question almost since then. I show this question almost every time in my talk. So the question is a bit philosophical, but in recent years, people have been trying to approach this by this reverse engineering approach. So that is, we try to identify all parts inside the brain and to map out the wiring diagrams, like for the computer circuit. So if we can really do that, then maybe we can simulate the brain in the supercomputer, or even build a new type of computer that can work like a brain. So that's the dream of reverse engineering. But of course, the challenge is the enormous complexity of the brain. So it's this system spans multiple temporal and spatial scales. We know that a human brain contains about maybe 100 billion neurons, and each of them make thousands or tens of thousands of synaptic connections with other cells. So the total number of synapses, that's the functional unit of a basic functional unit in the brain, in the circuit, is about the order of 10 to the 15th. So what I want to emphasize here is that besides this sheer number, more importantly, the complexity is due to the huge heterogeneity. Not only the number, but also heterogeneity. And there are many, many types of synapses, and even the same type of synapse can have very different functional states. So those states have different properties and obey different rules. For example, like what Guo-Qiang mentioned, this spec-time independent plasticity. For this plasticity, we basically we found that the synapse can change its strength depending on the activity of pre and postsynaptic neuron, and the time between this pre and postsynaptic activation determines the direction and the extent of the synaptic change. If we look into the figure, each spot here is one experiment. It's one synapse after this paired stimulation for a given time interval. But what we can see that besides the nice fitted curve that people can use for models, there is a very big heterogeneity that we try to ignore, but it's always there. It's bugging me since I've made this figure. For example, at 10 milliseconds, we can see that some synapses can have as much as 100% of potentiation, and some other synapses may not change at all. And those are the same type of synapse, same excitatory synapse between two excitatory neurons in the hippocampus, and they can behave dramatically differently. So why is that? That's something I keep thinking about. And then later on in another experiment that shows also the heterogeneity of synaptic plasticity, that in this case, the two students of mine, Zhong Xiaofu and Xiao Tan, did experiment with optical imaging. That the original idea of this experiment was to show, to examine whether mitochondrial activity contributes to synaptic plasticity. They actually found that this mitochondrial activity is indeed important for converting the short-term change into long-term change of the synapse. But what I want to show this figure here is that it's when for this same stimulus with glycine chemical stimulation that normally people call this chemical RTP, but it does induce synaptic strengthening for some synapses, but for other synapses, it actually weakens. So in G, you can see that it becomes two groups. Some synapses get potentiated, some synapses get even perhaps depressed. So that's another example of heterogeneity. So it's the synapses are different, even though we thought they should be the same, but they behave differently. And this is not totally surprising because synapse is complex. It has a lot of complex molecular machinery that can configure themselves into different configurations, and they may behave differently. So conceptually, this is acceptable, although we try to ignore them again because it's difficult to study this heterogeneity. So you can see even more complex. Within each synapse, there are thousands of protein molecules that interact with each other. So what I really want to do is, after a while doing electrophysiology, patching cells and looking for rules of synaptic plasticity, I realized that maybe we really want to see how synapses look like. So this is the reason so that I want to really see how the molecules in the synapses organize, and then to try to understand try to see whether we can understand how the synapses behave based on the molecular architecture. So how to do that? And this is Confucius told us more than 2,000 years ago that we have to sharpen our tools. And Sidney Brenner has a more precise saying that progress in science depends on new techniques, new discoveries, and new ideas. And he said even probably in that order. So I, because of this, I decided I should find new tools to look at synapses. And this is about 10, 15 years ago when I moved my lab back to China to USTC. And at that time, we know that electron microscopy has the highest resolution to see the macroscopic structures. And people use EM to characterize excitatory and inhibitory synapses. And we know that excitatory synapses usually are formed on dendritic spines, and they have thick postsynaptic density. And inhibitor synapses often forms on dendritic shaft, and they don't have obvious postsynaptic density. But conventional EM is still, as we can see here, it's not enough to really visualize molecules. We can see organelles, but we cannot see molecules. And at that time, cryo-electron microscopy has started. It's not as popular as today, but I do have a few friends working on cryo-EM, and I have luckily have two brave graduate students who want to enter this field. So they learned cryo-EM techniques from my friends, Peijun Zhang and Hongzhou in Pittsburgh and UCLA, and then we start grow neurons on EM grids, and then plunge that into a liquid nitrogen temperature to freeze them quickly to preserve the native structure. And then you under this cryo-EM, this is cryo-electron tomography. So the EM cryo-EM operates this single particle mode and cryo-ET mode. So at cryo-ET mode, we take projection views of different angles of the sample. So like X-ray CT in the hospital, we can reconstruct with computer the three-dimensional structure of the sample. And in this case, we can see the synapse from different angles, and we can see vesicles and many things. After we do this computerized reconstruction, we see much cleaner version of the 3D structure of the synapse. And you can see many details, including synaptic vesicles and ERs, and those are microtubules and even molecules within the synaptic cleft. So this is a typical excitatory synapse. As we can see, the postsynaptic, there are something density in the postsynaptic side. Also, this synapse forms on the dendritic spine. But we also see some synapses like this. This is also, you see postsynaptic density, and you see a lot of fine structures and ribosomes and microtubule. And interestingly, this microtubule actually come across this postsynaptic part. So this is not a span at all, so because span, microtubule cannot enter dendritic spine. So we see this, this has to be a shaft synapse. So this way, if you remember, I mentioned earlier that people usually say a shaft synapse is an inhibitor synapse. But the inhibitor synapse doesn't have postsynaptic density. So what kind of synapse this is? So to really identify this, we try to combine optical imaging with EM. In this case, we build a cryo chamber to put the sample in the cryo state in minus 190 degree and put that on the fluorescent microscope. And then the sample after fluorescent imaging, we can transfer the sample into EM. And that's what we can see. We see fluorescence imaging and also EM. And in this case, we can label the synapse with fluorescent molecules. And for this, we labeled, we transfect the cells with GFP labeled PSC95, that's a excitatory synaptic marker. And then also red fluorescent protein, Amchari tagged Gafrin, that's inhibitory synapse scaffolding protein. So basically in this graph, all the green dots indicate that this is probably an excitatory synapse, and the red dot indicates this is probably an inhibitory synapse. And we do see distinct features in the postsynaptic density for these two types of synapses. So this way we can identify excitatory and inhibitory synapse, and we can image them under EM. And this is a close-up view of an excitatory synapse. We can see the synaptic cleft, and we can see many particles or filaments, and those are probably receptors and other protein molecules on the postsynaptic membrane. And we can classify them. Actually, we can find a cluster of these particles and average them, and the shape of this can be fitted well with an AMPA or NMD receptor structure. So this is an excitatory synapse, and then this one is an inhibitor synapse. It has a typical thin postsynaptic density, and we can also see a lot of particles on the postsynaptic membrane. If we classify those particles, we can there is a distinct cluster, and this the average of these particles look very much similar to the crystal structure of the GABA-ergic GABA receptors, GABA-A receptors. So this way we can sort of tell ourselves that we do with cryo light electron microscopy, correlative light electron microscopy, we can identify and visualize the fine structure of excitatory and inhibitory synapses. And then we can further, at higher resolution, we can even see individual molecules in those structures. And they, for example, GABA-R receptors. So then the exciting perspective of this approach is that we now can see individual molecules. We can further study how they behave in the synapse, right? How they organize themselves into certain patterns in the synapse. So what we do is we choose a GABA receptor to test whether we can really study the organization of those molecules. And the reason we chose GABA receptor is not only because we can see them, but also because these receptors and their underlying scaffolding molecules are known to have, are believed to form some very elegant structure, like this hexagonal lattice that's for the Jeffreys scaffold. And they, because of their biochemical property, people have predicted that they should form this hexagonal lattice. And on top of the lattice, there are specific slots for GABA receptors to bind on. So if this is the case, then we should be able to see a very orderly structure of this orderly organization of GABA receptors on the postsynaptic membrane. That's what we were looking for. And but to do this, we need to find individual receptors. And initially, in our first paper, journal science paper, we did this manually. We just used eye to identify GABA receptors and then render them. But that's too tedious. So Xiaokang, Zhang developed an oversampled method that to do automatic classification. And with this classification, there are a lot of noise classified out, but then there is this one very specific structure that looks like a receptor. So eventually we can do the refinement and find the structure of this average structure of this particle in situ. And indeed, the structure of this class of particle is matched very well with the structure, the known structure of the GABA-R receptor. There are some additional things that we believe might be real because the purified structure was done cloned in this cell line. It's not in neurons, so they may have some different arrangements in their glycosylation and other modifications. So we think maybe we have the real structure of the receptor in situ, and the structure, the average structure we get is about 19 angstrom. So it's not very amazing. It's a lousy resolution for structure biologists. But for us, actually, I think the more important thing is that with this way, we were able to identify almost every GABA receptor on the synapse. So then we can really study the distribution of the synapse and the organization principle of the synapse. But from here, we don't see any a nicely ordered structure. Yuntao did some interesting analysis. So it did a simple analysis is to count the near neighbor distance. And he found that very interestingly, there is an 11 nanometer peak. And so that means that these receptors like to have a neighbor that's 11 nanometer from it. And if you pick a lot of receptors, near neighbor receptors, and average them together, you can see those structures here that indeed indicate that these receptors has something to link them to make them have 11 nanometer distance. So that's nice. That seems to fit with the idea about this lattice structure of GABA-R, of Jeffrin, and then the orderly structure of orderly organization of GABA receptors. But then he found that when one receptor links to two other receptors that also both are 11 nanometer, but then this angle seems to be variable. It's continuously variable, so it can be any angle. So that's very surprising. So that means although the distance between near neighbors are fixed, there is a large flexibility among them. And we have identified one receptor linked to 1, 2, 3, 4, 5 other receptors. So these receptors can link to other near neighbors, but this angle is quite flexible. So we can put pull all these linked receptors that has a near neighbor, 11 nanometer near neighbor together, and we can they can kind of form this we call this network. And the size of this network, the interesting thing is that the size of this network seem to follow some polar law distribution. That so you tend to have more than random larger networks. So that means that this organization is driven by some self-organizing process. So Yunta also did some entropy analysis, the Voronoi entropy. That's if you know this graph, it's a very simple way to assess the orderness in this two-dimensional structure. So he found that this entropy also is smaller than the random distribution, but it's not still it's much larger than expected from the crystal structure. If it's a liquid, if it's a two-dimensional crystal, it should be very small. But if it's a hexagonal lattice, it should be less than 1 or 0.5. But it's actually 1.5 something. But if it's truly random, it should be 1.7. So we call this a semi-ordered mesophysics organization. Then when we look at this further, we found that if you draw a boundary, you can see there is a pretty sharp drop. This organized linked receptors seem to form some condensate that put them together. So and they have a pretty sharp boundary around this condensate. And underneath this receptor, this is a layer of a postsynaptic scaffolding molecule. You also see some density that indicate that there's something that organizes here, so that supporting this receptor structure. So, but this is the interesting thing is that this boundary seems to be pretty sharp. That indicate that although the receptors can, there are some these solitary receptors that doesn't have a near neighbor that can be inside or outside the boundary, but the links of the receptors have this sharp boundary. So they tend to be that indicating that they are probably less mobile. Later on, we also try to link this with the scaffolds, and also we can see that very often you see each receptor has sometimes have a very clear postsynaptic scaffolding like molecule beneath it. So this is and on the presynaptic side, there are also vesicles that can be in this membrane contacting mode or in this tethered mode. And we found that the tethered mode vesicles seem to distribute it more uniformly within or outside the boundary. But this contacting vesicles seem to be more primarily located within the boundary. So that actually indicates that if these are vesicles ready to fuse, they seem to be able to dump their transmitters towards this condensate of receptors. So that gives some functional relevance of this organization. So to quickly summarize, we can see that with this method we can find individual molecules and look at their organization, and we found this organization is actually hierarchical. So there is this 11 nanometer interreceptor distance that link some receptors together, but it's also flexible. So when one receptor links to two or more receptors, their angles, these linkage angles are flexible. So that means this organization is semi-ordered. It's not truly. It's not like the lattice, but it's some kind of some flexible network, like we call this a mesophase. And it seemed to be functional and maybe relevant to some plasticity behavior, because this way we can imagine that with this semi-ordered organization of the receptor, you can disperse or condensate receptors quickly underneath the vesicle, then you can increase or decrease your transmission efficiency. So now let's. I think I'd like to move on to the other end of the scale. That's the architecture of the brain, and people have been talking about mapping the brain these recent days. And I think a couple of days ago, the Brain Initiative, BICCN in the US, just published 16 papers about the brain map in the mouse brain, and all this requires high-speed imaging. And the conventional methods, if you use the conventional histological methods, cut slices and then image with the conventional microscope, it usually takes months to finish a single mouse brain. So this is unbearable, although we have done that years ago. And but about 10 years ago, people have been developing this more efficient methods. So there are 3 examples shown here by in Pavel Osten's review. One is called serial two-photon tomography, and the other is called MOST. It's that imaging on the edge of the nerve. And the third one is called it's a clearance of the whole brain and use light sheet microscopy. And the first two-photon tomography and MOST both have reasonably high resolution and take about a week or so to finish the whole mouse brain. So this and the last method with this whole brain clearing and light sheet imaging, the resolution is not consistently high. So it's not really. It's used for some low resolution imaging, but cannot be used for fine scale brain mapping. And so this kind of connectivity map or projection map was primarily obtained by these 2 techniques. That's the mainstream right now for in the field. But still, even with those automated fast imaging techniques. It's still too slow if we want to image larger brains, like a macaque monkey is 200 mouse brains. If a human brain, if you want to image a human brain, it's 3,000 times mouse brains. So 1 week to image this is still too slow. A few years ago, we began to develop this new methods. It's kind of a hybrid approach that combines thick tissue clearing and a new method of fast 3D imaging. And this method, as we can see, this is the real time. The mouse brain was cut into 300 micron slices, and we can during the imaging, this sample moves continuously underneath the microscope. So with the conventional 3D image, you have to image take a 2D image at one field of view and then move to another field of view to take another image. And this, we realized that actually taking image, the data acquisition itself is fast. It takes a few milliseconds. But moving, accelerating and decelerating and stop from one location to the next, no matter how close you are, it takes hundreds of milliseconds. So most of the time was wasted during this movement. So Qingyuan and Zhu in the lab come up with a good idea that what if we image without stop? We just continuously image without stopping, then we can save more than 90% of time. So it can be 10 times or 100 times faster. But this continuous imaging when the sample is moving has a problem is because the sample, you get a blurred image. So we tried several ways, including deconvolution, try to solve this. But then one day, it doesn't work very well. But one day I realized that we can we can synchronize the scanning of the scanning illumination of the scanning beam with the readout of the camera, so that basically for every image, each pixel was scanned only once for a couple microseconds. And that way, there's no motion blur for every pixel. So this way, we get this high speed, we get very high speed imaging and of each slice, each 6 slices. We got 3D imaging for the 300 micron slice, and put them together, we can get the 3D structure of the whole mouse brain here. And this mouse brain was taken within an hour and a half. So compared to a week or 3 days, this is a big improvement in speed. And you can see the fine details inside this method. You get micron resolution or submicron resolution, and you can see the structure, fine structures of dendrites, and even see dendritic spines. So with this image, we can also reconstruct the brain and register that into the template, into the brain atlas. And then we can do not only structure imaging, but also the activity trace, like by CFOS fluorescence. And because CFOS is an immediate early gene that only expressed when the cells are active. So with CFOS expression, we can monitor which neurons are active during a period behavior time. So in this case, this is here we have two mice. One is a control mouse, and the other was undergoing forced streaming an hour before it's being fixed for imaging. So we can see that the streaming mouse indeed has more CFOS expression overall. And when we analyze 16 mice, 8 of them were under forced swimming, and the other 8 were controlled mice. We can see that the swimming mice indeed have more activation in those brain areas. So we did the whole brain analysis, but then we picked 14 areas that are related to, most of them were related to emotion. And we can see—
Which brain area?
So those, most of them were subcortical, that this is medial amygdala, central amygdala, basolateral amygdala, those hypothalamus, lateral hypothalamus, etc. So this line, each column is a brain area, and each line is a mouse. So we just choose these 14 areas, and we can see that firstly, indeed, the swimming mice have more activation in many of those brain areas. But then if you look more carefully, we can see that pretty much every mouse is different. So in terms of the overall activation, they don't behave entirely the same. Actually, the heterogeneity, individual variability is huge. So that's kind of a little bit surprising to us, but still, it's after we see the result, it's not too surprising, because we do know that animals behave slightly differently, and it's not surprising that they feel differently under the same stress. But in any case, this kind of surprising result indicates to us that if we really want to understand the animal behavior, we can't really rely on a few animals, right? So because they can give you a huge biased, largely biased results. Then with the fast imaging advantages of VISoR, 2 other talented, very dedicated graduate students, and then later postdoc and Fang is now an associate investigator in VIET. So they try to expand this technique from mouse to monkey brain. And this is about 300 times larger. And they have to image this monkey brain, actually they have to solve a lot of other issues besides imaging, because the monkey brain, when they cut them into slices, they tend to fall apart. So they have to develop a new embedding approach, and also some new clearing approach. And as we can see, this clearing is actually quite interesting. So they develop a new method called pure-clear. This primate, primate optimized uniformly clearing method that's different from the clarity method developed by Carl group that we also use for mice imaging. And this pure-clear is also important for monkey. It's also important because clarity doesn't work very well for the white matter in the monkey, because the monkey or primates have much thicker white matter. So it's with the regular clarity method, it doesn't clear very well. So with this pure-clear, this white matter gets cleared very well, as well as the gray matter. And also with neural trace staining, it's a gold standard of histological staining that pure-clear also preserves the cell morphology much better than this clarity. So we can see some images with the monkey brain that stained with this niso stain, and also with antibody staining. So this is the anti-GFAP that labels glial cells. So we can see this nice layered structure of neurons and glial cells. And we can see another, this is in the, I think this is in the cerebellum. You can also see exercise glial cells and neurons. And so This one, they imaged an entire monkey brain that labeled with 3 different colors. One is nuclear staining, DAPI, and then and the other 2 was a green fluorescent virus that labeled left, I think the left superior colliculus, and then the right fluorescent protein labeled the right side of the superior colliculus. You can see that, we can see that slightly later. Okay, here now you can see. So with these 3 colors, the microscope we had at that time had only a single channel. So for 3 colors, we image 3 times. Each time take about 100 hours, less than 100 hours. So within a month or so, we finished the 3 color imaging of this entire monkey brain. And the total data size is 747 terabytes for the 3 channels. And this brain we imaged we labeled superior colliculus, and the projection of those neurons was primarily confined to subcortical areas. So in the other brain, in the other monkey, we actually labeled a different location by accident. It's the MD, the middle dorsal nucleus of the thalamus. And this is happened to be a very interesting location that it's a main source for prefrontal projections. And what we can see that the neuronal fibers projects to the frontal area, cortical areas. And we can see the details of these sections, and we can zoom into this and see the fine fiber projections and abbreviations of the axons from those thalamic neurons. And here we try to map out this projection from this MD, and also some leakage to some small neighboring areas. But overall, most of these projection targets were MD projections. And we were also able to analyze individual fibers, because with this approach, when the fibers were not too dense, we can identify them. So in this case, we analyze the orientation of the fiber, use color to code orientations. We get individual fibers, all the individual fibers color coded in this neurons, in these projections, and we get this kind of somehow reminiscence of the DTI image of the brain, but the difference here is that each single fiber is a true single fiber, and so the orientation is a true orientation. It's not the inference from from the low resolution images. So I think we can eventually use this as a ground truth for some DTI image maybe. And we were also able to analyze individual axons in many different areas. Here is in this critical fold area. We just segment out individual fibers. We see many intriguing projection rods of those fibers. So Dr. David Van Essen saw this, and he was very excited. He said that it's basically counterintuitive for anatomists. So he said this maybe he need to rethink about this wiring principle. So what we can see here is that there are several different types of routing. That some axons can go from the white matter directly to the target. Some go this sharp acute angle turns, and some go this rectangular.
This is all following the surface, or what is it trying to follow?
We don't know exactly. So these are all thalamocortical projections. So most of those fibers come from the MD of the thalamus, and this is near their cortical targets. But they have to go through the fold. So they take different strategies of going through the fold. Some goes directly, some goes in first, and then go around it. I guess it may
folding of the surface, folding of the surface.
Yeah, the folding of the cortex. Yeah, the folding of the fold of the cortex. So that we can see this more clearly here. This is the cortical fold.
Because they cannot penetrate through the layers, just go through the fold. Okay.
Right, they cannot penetrate through the fold, so they have to go either below the fold, or they go into the fold, into the cortex first, and then go along the fold. So we don't know exactly what they mean, but they have different ways. May indicate that these neurons may have different functions, or they may have the same function, but just happen to have different routing strategies. And last, we try to trace the entire axon. So this is a very challenge because the data size is so big. It's hundreds of terabytes for the whole brain. So Lu Feng developed a smart method that basically cut the image into small pieces and then dynamic load the image required for the tracking, and it's very efficient. So he was able to track individual fibers semi-automatically, semi-manually, semi-automatically. That maybe a person it takes a person half a day to 2 days to complete a single fiber, and this single fiber is tens of centimeters long. Although it's maybe half a micron thick, but it's tens of centimeters long. So they were able to trace a couple dozen of axons from the MD to the frontal cortex, and here you can see one single fiber, the journey of one fiber. From the thalamus to the cortex. So I like this movie because it reminds me a little bit like Star Trek or Star Wars.
And this is kind of with almost near zero error rate when it goes along the track, yeah?
So we don't know exactly how to define the error. Maybe it makes errors, but it doesn't care. But in the end, the majority or most of the neurons they have to function correctly. So I guess you can say that it's error-free projection. And so this is pretty much all. We have this VISoR method. It's very high throughput, and it can be used to image brain structures, circuit connectivity, or neuronal activity trees. And it's very flexible. It's compatible with many different labeling techniques. So you can use it's very friendly for anatomists. And it can also be used for genetic labeling and viral labeling. And the more important thing for us is that it's scalable. So it can image as small as a mouse brain and as big as a monkey brain. And eventually, we hope that we can use this to image a human brain. And this is all the people who in the lab, past and current members, and I think the both people did the work that I've presented today, and we have a collaboration with Paming, and of course, and also Hongzhou at UCLA, and Peijing Zhang, who was in Pittsburgh and now in Oxford. and funding agency, funding from the Academy of Science, NSFC. This is the Ministry of Science and Technology of China, and also we have funding from Brain Initiative funding from NIH, and so I think I want to spend a couple of minutes to show the new challenges. So now we can image these brains. We can image a mouse. It gives us a few terabytes of data. We can image a macaque monkey, gives us less than a petabyte of data. And we can also then image humans, probably give us 10 petabytes of data. So it's a huge amount of data that's totally beyond our imagination a few years ago, I guess. And so this is really the real challenge is how to really understand this data, to analyze the data, to understand this data, and to convert from data to knowledge. So that's the real challenge. And I also think this is a great opportunity for interdisciplinary collaborations. I hope that's also the point of presenting the work in theoreticians. And we also, in Shenzhen, we try to build this center for interdisciplinary center for brain information, and we try to combine new techniques of imaging and electrophysiology and with modeling and computation, try to develop technology, obtain data, and drive discoveries and new theories. That's our hope. And we welcome everyone to visit us and to join us. And thank you very much.
Thank you. This is fantastic. Yeah, thank you very much. Are there any questions? People should maybe just raise. Okay, Peter, raising hands. Okay, Peter, I see a few hands raising. So, okay, Peter, you can ask your first question.
Great. Thank you very much. That was a fantastic talk. Really lovely, wonderful data.
Thank you.
So for on the synapses, on the cryo-EM, so you framed that by asking about why there's so much variability amongst the synapses. So I wondered if you have some in the plasticity, for instance, the different plasticity rules. So I wonder if you have any insights from the cryo-EM as to why you saw such variability. Have you discovered some of the rules behind that?
Yeah, so we actually, this is just the beginning, so we haven't really got much yet, but there are some observations. So the first thing is that is this organization of the receptors that we do see in this inhibitor synapses. We saw this semi-ordered organization that may indicate some potential mechanisms underlying this underlying plasticity. But we also see some heterogeneity in the postsynaptic density in excitatory synapses, and we haven't been able to quantify them very well yet, and we in some experiments we haven't been able to do like hiding plasticity type experiment like STDP, but we were able to do some homeostatic plasticity experiment by putting some synapses or culture in TTX for a couple of days. So they have changes, and those synapses do have some very interesting changes. And one very obvious change was that those synapses after being exposed to TTX for 2 days, they have much more dense core vesicles accumulated at the synapse. And they also have different organization of this synaptic vesicles, the clear, small clear vesicles, and that for transmission, the organization seems to be different. That they also different for excitatory and inhibitory neurons. So that basically the excitatory synapses have fewer docked vesicles, and inhibitor synapses have more docked vesicles after TTX. So that's consistent with homeostatic plasticity. Although I think in the past, most people studying homeostatic plasticity also focused on the postsynaptic part. And with EM, we will easily see the presynaptic changes. So that's some additional magnetic insights. But in terms of rules, we haven't been able to get anywhere.
Thank you. Thank you very much.
Thank you.
Okay. So, Seven, do you like to ask your question?
Thanks. Thanks for the great talk. I have one question. In one of your slides, you briefly mentioned about the structure of gliases. I was curious if you also pay attention in the course of your investigation to the gliases, and if you observe any kind of characteristic patterns in the gliases also in the brain.
Oh, we for that slide, for now, we haven't been able to systematically investigate glial cells. It's just we use that just as a very very preliminary pilot experiments. We just show that the technique works. We were able to see glial cells in the whole brain. Currently we are doing some experiments regarding glial cells, changes of glial cells in learning, but we haven't. It's at the very beginning of the experiment. We haven't been able to get anything yet. Thank you.
Thanks. Thank you. Hopefully next talk.
Yeah.
Yeah. Thank you. Yeah, so the advantage of this method is that whatever you want to see, you can look at that. As long as you can enable it, you can look at that in the brain-wide scale. You can see the whole brain instead of just pick a area to study. So this way we don't make biased presumptions of which area may be important for certain things.
Thank you.
So in one of your slides, you have this monkey brain, I think it's monkey brain, stained 3 different stains, and then you scan them. This is not simultaneous. One particular color you scan once, another particular color.
That's right. That's right. Yes. Yeah.
And but maybe I misunderstood. So because you need to slice them into slices. Yeah. So it's the same brain. It's stained once.
It's the same brain. It's the same brain that can be stained for multiple colors. Yeah, I think I didn't state clearly. It's ideally we can image all the colors at the same time, but yeah. But the setup we had at that time has only one channel. So
so it's you zoom onto one particular color at a time. So
no, we use one laser, one camera to image one color through the whole slice, and then it change the color and image change the laser and image to turn on the other laser and image another color again and then again. So but it's the method, the system is stable enough. So this 3, even though
registration is still there. I see.
Yeah, the registration is easy. It's relatively easy, so it's not too much.
That's why you could do that. Even though it's already cut into slices, as long as these 3 terms, the physical registration can go back, you can still reconstruct the whole thing. I see.
Yeah, so that's for color, you register the 3 colors within each slice, and for registration for stitching all the different slices into the whole brain, that we need some alignment. So that actually takes some deformation and all this manipulations to align them well. So we were able to do that with reasonable accuracy, that about 5 to 10 microns accuracy. But for single fiber tracing, you can stitch, you can basically correct all the misalignments to a single fiber resolution. That's also
okay. Okay, I see. And also, I wasn't completely following with this GABA receptors. You say they kind of form this kind of, you know, some are they're 11 nanometers apart. Yeah. That's a favorite distance, but some are 1-on-1 connecting, some are 2x2 or 3x3, and all this, and therefore, let's say by n, they have a much bigger network and a smaller network. Is there any functional thing you maybe connected with that? If the one single synapse has all this thing, or what is it? Yeah.
Yes. That's very good question. It's I think it's. So the observation indicate that first, the receptors are indeed, many of the receptors are indeed linked to other receptors. But secondly, this linkage is not a rigid linkage. It's not like there is a hexagonal lattice formed beneath it. So it's a flexible, it's more like a flexible net and also there are also receptors that are not linked to any other receptors. So that indicate that this is a mixture, maybe a mixture of gel and soluble components. So it's or it's something similar to this it's getting popular now. It's the liquid phase separation that they, because of their multivalent interaction, they tend to form condensates. But here, I think it's more than that. There are this component of this multivalent interaction that form condensate, but there are also some stronger, some more stable interactions. So we don't know exactly how, but when we simulate, we actually did a simulation, just try to put all the interactions between the receptor and scaffolding molecule, between different scaffolding molecules together. They can eventually form this kind of network and have similar statistical properties.
I see. Jingyu, were you raising hands to ask questions? Do you like to ask a question, Jingyu?
Yeah. So actually, I was wondering, as you say that the fibers that you track in the brain cannot be too dense, like cannot be, if I understand correctly, cannot be like too close to each other. But we know that many fibers are like form a fiber bundle in the brain. Especially for those long distance connections, so used to track the dense kind of fiber.
I guess what I meant was technically, if they are too close, if you have two fibers both labeled, and if they are too close to each other, we can't resolve them. So we can't really get single fiber resolution for those samples. For that purpose, we have to do sparse labeling in order to solve individual fibers. But you're right, it's indeed in the many axon fibers tend to grow in bundles. So that's something it's something we want to look into more carefully in the next step.
So I mean, essentially, in the end, maybe with some correct analysis, you can track almost 100%. I mean, all the fibers in the brain.
The resolution limit makes it basically impossible for this optical methods to track 100%. If you label 100% of the fibers, then you cannot resolve much. With EM resolution, for example, like Jeff Lichten did on the small piece of mouse brain or small piece of human brain, you can trace individual membranes, right, fibers through the EM 3D EM image. But but it's the image is too low throughput, right? So you spend a few years to get 1 cubic millimeter. So that's for the large scale. So I guess it's complementary. You have to use very high resolution and try to get the complete connectome for a small piece of sample. And in the meantime, you also want to do sparse sampling to get the overall picture for the large scale. So that's we are doing this sparse approach to try to second approach. I guess for this statistical consideration is very important. How we can derive rules or principles from the sparse sampled data. That's a real challenge. Also.
Thank you.
Very interesting. Any other questions? Please raise your hand. Yeah. If not, I am also wondering. You have these fibers that going along the fold, the inside fold. So for instance, if one fiber is going this and folding to that, but its destiny is actually there. Yeah. So if it doesn't go this and that, but it wants to go folding to fold and there. Would that mean that it's trying to also connect something there? Or
maybe, but we don't see branches. So we do see branches. We do see branches elsewhere, but we didn't see branches for those fibers that I showed.
So someone goes in there, turning and go.
Yeah. They seem just different routing strategy for them. There are I think 5 or 6 classes of this routing strategy. It's not totally random. It's not random. It's they fall into one of those classes.
Interesting. So therefore, it's a little mystery so far, but nevertheless, there must be either.
Yeah. How come that they can develop those? It seems to be also precise because many different, not a single fiber. It's just a bunch of fiber following.
Going like that way, yes, yes.
Yeah, and they always go correctly. So there may be some cues for the guidance during the development.
They have some guidance mechanism.
Yeah.
Okay. Or maybe it's growing when it's developmental stage when the brain is not folded, like a marmoset kind of brain, and then when it's folded, it kind of relaxing too.
Some of them maybe due to that, but that's
interesting. Any other question? If not, this is actually quite late for Professor B because this is really 10 o'clock, past 10 o'clock in So thank you really very much. We're going to have some one-on-one meetings, but not so late for you. So we do it. Really, really fantastic. This is kind of even though we have COVID and we cannot really see each other, so it's fantastic you have time to give us this late. And we hope to have I look forward to meeting you tomorrow, have a further talk, and we hope to have a future chance for have you to come here, and I love to visit with you. And thank you so much, very much for your.
Thank you. Also, really look forward to come to Tübingen.
That would be great.
Several years ago, but I really want to go again. And also, all of you are welcome to visit Shenzhen or Hefei.
Yes, I was hoping, but down it's a little. So I had to wait a little bit. Maybe with the travel. There's still like a 2 weeks travel, but I don't want to talk about this now. 2 weeks of a quarantine, but hopefully that will be removed. I hope the world is going forward.
Next year.
Great. Better. Thank you so much, really, and so have a good evening. Thank you. Okay, bye. Let's thank Professor B again. Thank you very much. Thank you. Okay.
So just one more thing. So for the individual meeting will still be within this Zoom link. So
within the same Zoom. Okay. Thank you very much. Thank you, Jingyu, for all this organization. Thank you. Okay. Bye.
Thank you, Jingyu.
Thank you.